Welcome to this video in the Blueprint of Life topic. This video is going to be looking at the syllabus dot point, explain how mutations in DNA may lead to the generation of new alleles. A mutation is a change in the DNA information on a chromosome, or more simply, a change in a gene. It can result in an alteration to the original DNA sequence or to the actual chromosome number of the organism. When a mutation occurs in a sex cell, such as our sperm or our eggs, the changed genes can be inherited. Many inherited mutations are lethal, so in other words, the offspring will not survive through the length of the pregnancy and therefore will not be born. In some cases, the inheritance of the mutated gene results in the acquisition of some diseases, but in rare instances, the altered gene may in fact result in beneficial characteristics. This can in turn produce more variation within a population and so increase the chances of evolution occurring within a species. Most mutations occur while the DNA molecule replicates. As we know, a gene is a segment of DNA that provides the instructions for making a protein, and proteins influence our characteristic. This table describes how two different versions of a gene can result in either normal skin and hair color, or albinism in some people. So we can see in our first row here, we have the version of the gene that provides instructions to create a normal protein enzyme is translated or undergoes a process of protein synthesis to produce a normal enzyme that makes the pigment molecule in our skin and our hair, and therefore the characteristic that we see is normal skin and normal hair color. However, in this bottom situation, we see that the version of the gene that results provides instructions to make a defective gene. So through the processes of transcription and translation, we have the production of a defective enzyme that does not make the pigment molecule necessary. So the characteristic that we see in individuals is albinism, and these are the individuals that have very pale skin and very light colored hair. There are a number of factors that can cause mutations, and these are known as mutagens. These can include several forms of radiation, including X-rays, ultraviolet light, and gamma rays from nuclear reactors. Some chemicals also act as mutagens. Examples can include asbestos, certain dyes, nicotine and benzene, which we know are found in cigarettes, and some preservatives which are found in food. In a small number of cases, mutation may occur spontaneously, and these are known as background mutations. So along with different causes of mutations, we also have different types of mutations. The first one we're going to look at is changes in the chromosome number. In some meiotic divisions, chromosome pairs may not completely separate. This is called non-disjunction and results in the gametes either having too many or too few chromosomes. Down syndrome, in which a child inherits 3 copies of chromosome 21 instead of only 2, is an example of this. So any individual who has Down syndrome, has ended up with 3 chromosome 21. Now, majority of the time, having too many or too few chromosomes is lethal, and the individuals will not survive. However, it has been found that those with trisomy 21, or 3 times chromosome 21, survive, and they have these very obvious features which we can see in this image of this little boy here. Another situation in which chromosomes fail to separate during mitosis can result in an organism inheriting more than one set of chromosomes, and this is referred to as polyploidy. Polyploidy is in fact a useful feature in commercial plants and is used by plant breeders in the production of hybrid species. So it helps to create extra flower buds, which therefore produces extra flowers and fruits. So therefore, obviously, in agriculture, where farmers are relying on the production of extra fruit for them to be able to sell that on, polyploidy is obviously going to be an advantage. However, in humans, polyploidy can result in things like extra digits or extra fingers or extra toes if the chromosome does not separate properly during mitosis for that particular chromosome. We also have mutations that result from changes in the DNA base sequences, and there are 4 different changes that can occur to the base sequence in a strand of DNA, and these can include substitution, insertion, deletion, and inversion. So in substitution, here an incorrect base replaces one of the original ones in the DNA strand. The result is that a different amino acid will become incorporated into the chain during polypeptide synthesis or protein synthesis, and a different protein will be formed. This may function differently to the correct protein and create enormous problems for the individual that has inherited the faulty gene. An example can be seen in the disease sickle cell anemia, where the mutated gene varies from the normal hemoglobin gene by only a single amino acid. Symptoms of this disease include the formation of sickle-shaped blood cells at low oxygen levels, which can block blood vessels. So sickle simply means it's more of a C-shaped than that nice donut shape that we had a look at in the maintaining a balance topic. So here we can see in this particular example, the base C from our original strand of DNA has been substituted with the base T in the new strand of DNA that has been replicated. And as we can see, that's resulted in a different mRNA strand and then a different amino acid forming in that space there. The next one is insertion. In this type of mutation, an extra base is added to the DNA sequence, again resulting in a difference in the amino acid sequence and the protein that is formed. From the point of the mutation onwards, the amino acid sequence will be completely different, causing what is known as a frame shift mutation. And again, the result is usually a non-functioning protein. So here we can see the original base sequence was CTT and then CTC, but here we can see the insertion of a new C base or a nucleotide containing a C, which therefore is transcribed into a G through mRNA, and then we have a new set of amino acids forming because everything shifts one over in our DNA strand, which results in everything shifting in our mRNA strand, which results in our different amino acids forming. The third type of changes in our DNA base sequence is deletion. The effect of this type of mutation, in which a base is deleted from the DNA code, is similar to that caused by the insertion of a base. So we can see here our initial sequence was AAAA, where in our new strand of DNA we have AAAT. So that A here has been deleted, been completely removed from the DNA sequence, and therefore now everything shifts one this way. So we then end up with a new mRNA strand and again a new sequence of amino acids. So we can see here that after the deletion is where the issues take place. And lastly, the last type of DNA base sequence changes that results in mutation is inversion. So here, due to a mistake during DNA replication, a whole base triplet is back to front. Again, this results in a completely new protein with different properties to the intended one. So here we can see that we had our original chromosome reading in the alphabet A, B, C, and then D, E, F, G, H, I. And what's happened is this middle gene has switched around and our new chromosome or our new DNA sequence becomes A, B, C, F, E, D, G, H, I. So this would obviously transcribe to a different mRNA strand again, which would result in a different protein being created. So this here is just a fun little image to help you try to remember the different types of mutations that can occur due to changes in our base sequences. So we start off with our original DNA, which is our beast. We substitute the B for an F and it becomes a feast. We insert the letter R and our beast becomes breast. We delete the letter A and our beast becomes best. We invert the last two letters and our beast becomes beats. So this is just a little bit of a fun way to graphically get your head around the different types of DNA changes that can occur and what they look like. And that brings us to the end of this video. So thank you for watching.